So greetings to all. Last week we had Dr. Andakeliu Meonge from Tari, Tanzania, presenting interesting research on sorghum and how the caste markers can be utilized in the breeding programs in the sorghum in Tanzania. So today we will be introducing wheat research from CIMMYT with the support of Kelly Eversole from Eversole Associates, United States. I think everybody know very well Dr. Kelly because she's very renowned leader in agriculture genomics, biotechnology, and pre-competitive public-private agriculture research. And since '94, she has spearheaded international consortia and projects aimed at obtaining reference genome sequences for critical crops such as wheat, but also in livestock and poultry species. Please, Dr. Kelly, correct me if I'm wrong. Kelly is particularly noted for her leadership in the International Wheat Genome Sequencing Consortium, which successfully acquired the reference sequence of the bread wheat genome. So since 2016, she has led the International Alliance for Phytobiomes Research, pioneering a holistic systems approach to understand the complex interactions between agriculture production systems. I would have to say much more. I have a really very big text. I invite you to see the website from the International Wheat Genome Sequencing Consortium, but also the Eversole Associates. But Kelly can have received numerous honors, including the Outstanding Performance Award from the National Corn Growers Association in '99. Kelly, thank you very much. We are very honored to have you here and to have your time. And I will let you add more to what I said and to present Dr. Susan to the audience. Thank you very much.
Thank you very much, Ana Luisa. It's a great privilege for me to be able to present today. I do have a few slides, but before I do that, I will say that you are right. I did lead the effort to sequence a number of the livestock and poultry genome species, to get sequences for them, and that did include the chicken genome. So which is an interesting genome by itself, and pales in comparison to the wheat genome, which had I known, I might have paused just a little bit before jumping into sequencing the wheat genome, given how large and complex the wheat genome is, as our distinguished guest who is going to speak to us today knows very well. But I will share a few slides just to give you some updates on what we are doing in the wheat consortium. I'm not going to be presenting any science at all at this point, but just a quick overview. So we were started in 2005 by a group of wheat growers and scientists, as well as public and private breeders. We have involvement of over almost 3,500 members in 76 countries, and who represent almost 1,000 institutes. We also have 8 sponsors who make all of our work possible, and I want to take a moment just to thank them and to point out Kansas Wheat was one of our founders, and they have continued to see value in genomics for wheat improvement. So I really want to call them out as being very supportive. And CIMMYT as well has been a sponsor for quite some time. So our main vision has been to enhance breeding through an increased understanding of the molecular basis of traits and their allelic diversity. And we really formed this vision after we published the first reference sequence. And the idea was, okay, now how do we use this reference sequence to really improve wheat? And so we revised our vision and really began to focus on molecular basis of traits. So our activities this year is we have recently received funding from the U.S. National Science Foundation, as well as NRA-E and the EU for developing platinum quality sequences of 12 land races, plus another platinum quality sequence of the reference species, Chinese Spring. And our goal here is to really represent the breadth of wheat diversity, to show the historical wheat diversity. And then we will also include automated and manual annotation and the development of a practical haplotype graph, which will enable all breeders, public and private, to actually compare their varieties with all of these different sequences. The practical haplotype graph will also include all of the publicly available sequences of wheat that are out there. So if you are working on a wheat genome sequence of any kind, and you will have a sequence of any quality by the end of this year, please let us know about that. We are also continuing collaborations with wheat genomic tool developers to try to make sure that wheat scientists have all the tools they need to actually use all of the genomic resources that we have. And we also have our own webinar series, and we are always looking for scientists to present. So please, if you have an interest in presenting your science during our webinar series, please let me know. We also will be participating this year in the International Wheat Congress and PAG, the Plant and Animal Genome Conference Australia. And we plan to have a call for abstracts for a PAG Australia workshop. So if you have an interest and you plan to be in Perth this fall and be attending the Wheat Congress or PAG Australia, submit an abstract and you may be selected to give a talk at the IWGSC workshop at PAG. We will start this summer planning for the 2025 workshops at the Plant and Animal Genome Conference that's held in San Diego. That'll be held in January. And we will usually we issue our call for workshop abstracts, I think in late July, early August. And we'll be calling not only for speakers we have 2 workshops at PAG, but we also will be calling for abstracts for early career award. This is a Catherine Fouillet IWGSC Early Career Award, which provides a $1,000 stipend for the recipient and allows the recipient to speak at the main IWGSC workshop. So those are some opportunities that may be available for scientists in Africa as well as other countries. So just stay tuned. You can find out information on the IWGSC website. I'll also just give you a link to our team. We have a really great team that's been with us for many years. I want to point out we have a new team member, Rolanda Young, who is our events manager. So you can, if you have an idea for a webinar, you can reach out directly to her as well as to me. So with that, I want to turn to the main event for today's webinar. It is a great pleasure for me to introduce Dr. Suzanne Delaizekaka, and she's the head of Wheat Genetics and Molecular Breeding Laboratory at CIMMYT in Mexico. It was a great honor for us this past year, for her to join the IWGSC Board of Directors, and her participation in our board really shows that we are focused on ensuring translation of genomics to application and to wheat improvement. Suzanne is well known in the community. She's a champion in wheat genetics research. Her research interest has been unraveling the genetic foundation of vital traits in wheat to advance genomics-assisted breeding strategies. And she is an extremely well-published author with more than 300 articles. So without further ado, I will want to welcome Suzanne this morning or this afternoon, if you're in other places, for being here today. So Suzanne, I will turn it over to you.
Thank you, Kelly. I will, if you can just stop sharing your screen so that I can share mine. Yeah, thank you.
Sorry about that.
Don't worry. Okay. So can you see, just a quick confirmation by everybody, because I have a small screen. Can you see my screen in full mode?
Yes.
Yes, it's in full presentation.
Okay, good. So, thank you. Thanks so much, Kelly, for the introduction. So in line of the topic of this seminar series, I thought I'd give a quick summary of how we, first a little bit about the Global Wheat Program of CIMMYT and how we deploy specifically the shared CG genotyping service in our program. So the Global Wheat Program has 3 breeding programs, the 3 wheat improvement programs. First, 2 programs are related to spring wheat type, bread wheat breeding program that is led by Leo Crespo and Velu Govindan here in Mexico, as well as a durum wheat and Tuitikeli breeding program led by Karim Amar. In addition to that, we have a wheat type breeding program also in Turkey. That is, the breeder here is Behan Ekin. So I'm mainly in my talk referring to really activities in the spring bread wheat program, just because it's the program that targets most of the area in the global south, so the regions we are targeting, what is has an overall 65 million hectares in diverse environments. So the main environment being under irrigated conditions, around 26 million hectares, mainly in the southwestern plain zone of India, central northwestern Pakistan, also Nepal, and irrigated regions in Ethiopia. So the spring bread wheat breeding program, similar to I think CG breeding programs, has more recently divided or restructured its targets. So we have in total now 5 different breeding pipelines that are distinguishing the target area really across the global south. So depending on the seed color really of wheat, white and red seeded, but also on the environment, what I just mentioned, really the irrigated or rather rain-fed condition, and also depending on the maturity type. So the latest pipeline 5, was just recently moved to Kenya in a collaboration with CIMMYT and National Institute Calvo, and is now led by Sridhar Bhavani. Just some quick emphasis. So we have some really core trait across these different pipelines. So they are important in each of these 5, and just to give you an idea, this is high and stable yield potential, end-use quality, but also nutritional quality. The main disease in wheat is indeed rust, and it's basically everywhere. And then we have specific traits that are more targeted to the different pipelines, such as Septoria triticale blotch, another devastating disease, or Fusarium head blight, or more recently wheat blast. So the impact of the program has been, I think this is well known, has been large. I put you here two different estimates. So the first one on the left side is from an impact study in 2016, where it was shown that in the time frame from 1994 to 2014, so really many CIMMYT or CG wheat germplasm was grown across the globe, with the main impact in the area of the South Asia, Sub-Saharan Africa, and also West Asia and North Africa. So recent genetic gain assessment done by the Global Wheat Program estimates an annual genetic gain between 1% and 2%, what is relatively high. So the breeding teams or the pipelines are well supported by different enabling units. So really want to stretch the point that this is an extremely larger team effort. So we have enabling units such as quality laboratory. We have our regional office that give us feedback how the lines are connected to the national programs and how lines are performing in the different countries. We have a physiology team, a seed health unit team. So all these teams are supporting the wheat improvement programs, and including the molecular lab that I'm leading, and what I'm now focusing a little bit more on. So the activities of the lab are really supporting the trait discovery and deployment activities. So we do support genotyping for trait discovery. We do marker-based or trait-based design, marker designs and validation. We lead and support marker-assisted trait integration and deployment. I will come back to that in a minute. We also support the genotyping for genomic selection deployment. I think we are leading the integration of these genomic tools into breeding, and often drive proof of concept projects of novel genomics assisting breeding approaches. So as I guess in each of the programs, this has been the integration of these genomic tools has been a continuous process. So it's a constant move and change of technologies and approaches. So this is the Global Wheat Program, I think, is linked to molecular work almost 20 years now. So starting in the very early in the centuries, where the focus was rather gene identification and biparental mapping, up to today, where we try to integrate more and more big data sets, rapid integration of genomic outputs, up to sequence data and new bioinformatic tools. So just a brief slide on the trait discovery research. I don't want to go as much in detail, but the program is structured as such that we have geneticists actually linked to our trait specialists that perform trait discovery research, mainly in the area of enhanced nutritional value. So we do some work on dietary fiber, for example, mineral content, zinc and iron, starting on the pre-harvest sprouting and also whole grain quality characteristics. Also related to biotic stresses, certainly WAST is here number one. Niela Kerishin in the wheat program is leading this, but also related to other diseases, what I mentioned already before. Wheat plus being high priority at the moment, also Fusarium head blight, but also others. And in addition, some work in two big consortium that the CIMMYT program is involved in, which is the IWIP, the International Wheat Potential Partnership, but also HEDWIG, the heat drought partnership, where we look at components of especially heat drought tolerance and yield potential. So just edit this slide because I want to stretch that specifically the trait introgression linked to molecular markers is not the only trait introgression pathway we have in wheat. I'm not sure if it's fully crop specific, but we also have other introgression pathways like white crossing, or strategic crossing, and population improvement projects that lead to the introduction of new traits and testing of new traits in the program. Molecular markers are used in a number of them, mainly in the rapid trait introgression of marker selectable genes or QTL, but also in population improvement, and if possible, when we introgress or have crosses with relatives. So specifically the rapid trait introgression of marker selectable genes or QTL, we did centralize this a few years ago, making use of some new facilities at our station in the south of Mexico, about 2 hours from our headquarters in Toluca, where we have around 2 hectares of a screenhouse space. Meanwhile, with some light improvement, and also 2 fully equipped speed breeding rooms. So what was the motivation to really centralize these marker-assisted trait introgression? Simply to have a better coordination across the breeding programs, to have a clear alignment to the established product profiles and market segments, also to have transparency of the trait advances internally, but also externally, so in collaboration with our national programs. Also to use exactly that speed breeding facility for faster delivery of these traits to the breeding programs and to our partners. And a last reason really to streamline the genotyping logistics and exactly the use, make best use of these shared genotyping services. So currently, how do we prioritize these the genes or QTLs we introgress? It's based on certainly demand and value. So we estimate the demand of the associated traits in each of the product profiles. So this is defined in the product profiles. Certainly the evidence of the QTL and its gene value that is documented. There needs to be a lack of phenotypic variation or genetic diversity of that trait in our program for a QTL to have attention. Selection efficiency, so sometimes the markers are certainly more efficient than the phenotypic protocol. Do we have available or sufficient accurate markers to use for marker introgression, and certainly available funding? So how we do this practically, we have a kind of advancement meeting every year where the trait specialist and the geneticist and the breeders meet to discuss the demands and the new candidates, selection candidates, and then we make a common decision what to move into these trait pipelines every year. So this is just a summary of the last 3 years. What has the genes that have been introgressed? So it's, as you see, many rust resistance genes for stem rust, yellow rust, but also fusarium head blight, so the same diseases as mentioned before, Septoria tritici blotch also, and more recently yeast blast. So what I highlighted here in orange are the genes related also to durum wheat. So this is in spring and bread wheat, and in durum wheat introgressions. And then we try to enhance a little bit. We know it's highly quantitative traits, but enhance a little bit the diversity with QTL. We have discovered specifically in in genomic resources, so wild relatives that we know they are not very frequent in our germplasm. So we also integrate some of these to a certain extent. Okay. So the integration is a relatively simple protocol at the moment. This maybe has room for optimization. So annually we have the capacity to do around 100 new crosses. We decided in the program to target new genes maybe every 2 to 3 years only. So we want to increase the integration in the diverse backgrounds. We do sometimes also F1 by F1 crosses to pyramid, specifically some of these resistant genes. So we have now 2 or 3 genes pyramids into the pipeline. So we try to integrate in around 10 to 15 different recurrent parents that are selected by the breeder, Velu in this case, depending on the demand and on the trait pipeline or the breeding pipeline. The trait is most important or most demanded. And usually we do 1 or 2 back crosses. This is done fully in the screen house and advance until F3, so early stage. And then it goes into the field, into Luca for observation plot, basically, sometimes already trait-based, so depending on the genes that is integrated, and then it goes into real yield testing here in Obregon. If you see, not sure if you can see my background picture. This was, I chose that on purpose. So this is a picture from 2 days ago. I'm currently in Obregon. This is a picture where we demonstrated some of these products to our visitors in the field. So in terms of, so here really in this trait integration pipeline, so we are fully supported and we fully build on the CG shared genotyping service, or the low density genotyping. So all the optimized production markers are basically run by Intertech. We send our samples to the lab in Sweden. So we build on the fast turnaround and really the low cost and the multiples of 4 plates that the lab can easily run. We have some issues because of the complexity of the wheat genome. So we have some markers that are really difficult to run. So we support the genotyping effort with our in-house laboratory. Also, we have still a few markers that are not on a SNP level. So we also support with our in-house laboratory these data types. In addition, in some cases, we have very small genotyping jobs, not in the framework of this trait integration, but more in the framework of research that we support in-house, and certainly we support all the new marker design and marker validation. Few issues we have just to see that you get an idea on that complexity what we have with the low density service genotyping. So wheat, as you know, is a hexaploid a genome, so it has 3 full genomes that are very, very similar in cases. So we struggle sometimes to really design markers that are very specific to one of the genomes, and that leads, as you can see here. So I put here some pictures where you can see this is the automatic calling from the SNP through Intertech. So what happens very often that the automated calling does not take into account the genome non-specificity of our SNP. So we often have to recall the SNPs manually after we get the data. Also in cases we find variable proportions of missing data. And right now we have a little bit of an issue, I thought I raised that, of the Scriptoria submission tool that is for us not fully operational yet and not linked to the EBS. So we are using currently 3 different systems to to order our genotyping requests. In terms of genomic selection, I thought I quickly describe what the Global Wheat Program has been changing in the there's been quite a change in the breeding scheme in the last few years. Also, a lot of movement in the program. You might be familiar with the classical shutting breeding approach that Norman Borlaug implemented in the 40s and 50s in Mexico, what was basically shuttling the segregating populations from the north to the south of Mexico, so 2 cycles per year for up to final yield testing here at Obregon, where we have a high yield potential site. And usually the breeding cycle was around 5 years long, so parents were recycled after 5 years, and elite lines or the best lines were distributed globally after 7 years. So this has by now relatively shortened by 2 years. So instead of having 2 cycles in the first years, the breeders are now able to run 3 cycles in the first year. So they cut here already half a year. Also, they go into spike selection 1 year early, and they go into testing 1 year early. I'm not going here much into detail. To conclude, so the parent recycling is now after 3 years, and the international distribution of the lines already after 5 years. So the genotyping related to genomic selection was built into these pipelines. So we are adapting, still adapting to the new pipeline. So genomic selection in the program started around 2014, funded by Feed the Future, USAID funded Feed the Future project in collaboration with Kansas State University. And initially we did genotype the lines that enter in our first stage yield trials. At that time, this was around 9,000 lines, which were annually, so for 5 years, genotyped, and the predicted values were provided to the breeders for additional selection support. So at that stage, the lines were only tested in one environment, 2 replications, so there's limited phenotypic information. From the platform that was used here is genotyping by sequences, and that was supported by the Kansas State University laboratory. So from 2020 to 2023, so the project decided to move 1 generation earlier. So this is what we call Parcela Chica, a PC stage. So this is the lines that will enter into our first yield trial stage. And at the moment, these are around 30,000 lines that are annually genotyped and are used for genomic selection. So what the breeders do at this pre-S1 stage is they use visual selection for disease, phenology, and height. And we do add the predicted values, the GBVs, but also values based on our pedigree information. We have a very good pedigree system in wheat, and that for probably more than 40 traits. So we predict for all the industrial and nutritional quality traits, but we also predict for disease traits, not only phenotypes, not only taken within Mexico, but also disease screenings on our historical lines that come from India, from Africa, or other countries from South Asia. And the breeders use these predicted values very heavily to select around 4,500 lines that go into the yield trials. Sparse testing at that stage is in planning. This is not applied yet. The same genotypic data are then used also to derive marker-adjusted phenotypes 1 year later, and they are shared with the national programs for parental selection. And we are also planning a more CIMMYT NAS partner genomic selection network, especially in the phenotyping network we have already in place in South Asia. So this project ended last year with Kansas State University. So this year, so we decided, as other CG centers, we have built, thanks to excellence in breeding, really, I think the initial low-density genotyping service is really a large success story in the CG. So I think the idea of building up a mid-density genotyping service was a great follow-up. So similar to other crops, we developed these mid-density DART tag panels. So in wheat, it has currently 3,900 SNPs, widely distributed. You see that at the pictures at the right, widely distributed across the genome, and it includes gene-based or trait-based markers, but also markers linked to QTLs. And in the last 2 years, we have really testing these platform forth and back for different purposes, and we constantly see that the platform gives us very similar, sometimes a little bit better, sometimes a little bit lower, similar prediction accuracies compared to the GBS data we were using up to last year. So we are pretty confident that we can easily move to this platform this year. So you see here below the correlations between the different predicted values are usually very high, between 0.8 and 0.9. I think the platform had also quite a good uptake in the wheat community. So it's used for genomic selection, but also for variety identification, general fingerprinting. I think ICARDA has been using the platform to fingerprint their gene bank accessions. It's also used for marker-assisted background selection. So we are still, this is still work a little bit, there's still work in progress related to this mid-density genotyping service platform, because the platform for bread wheat works quite so well for us. We decided with aim to design a platform also for durum wheat. So this is close to be tested. Around 3,600 SNPs are collected. In addition, we want to update the bread wheat platform, as we see some markers of that are constantly failing. So I think we can improve a little bit this platform still, even so it works quite well. Also, what we are testing is a mid to high density imputation option. So we're working with 2 different research groups on that. The first related to the practical haplotype graph. Again, we are collaborating with Kathy Jordan from KSU. So the US genotyping labs, they have built already a practical haplotype graph, what is basically a reference population of exome capture data of US and now CIMMYT varieties. So she's currently trying to impute the raw data of our DART tag panel to this reference population to extract a higher density haplotype map. And we want to see if this boosts further our genomic predictions. Similarly, we work with a group, DICA in Australia, that have an in-house imputation pipeline, similar to really a reference population of exome capture sequence data. And this is ongoing. The population is a little bit higher or bigger. They have up to 800 different exome capture sequences or genotypes that are exome capture sequence. And we already had first success, at least in aligning Australian genotypic data with our GBS data. So this has been working right well, and we want to extend this now to the DART tag panel. Then we started to work a little bit, something like what we need, something like a haplocatcher. Currently trying to develop some in-house scripts. So what we want to extract really from the DART tag panel is a postulation of of the presence of the genes we are targeting. I think that would be super useful for the breeders to get a side report in addition to the DART-TAC marker report that postulates the different genes and can validate these genes with the phenotypic data on the same data set. So to basically verify specifically for resistant genes, or to constantly verify still the function of these genes in our germplasm. So it's something we want to work on. And if somebody is doing in the audience working in that same direction, I would be really happy to start collaborating more deeply on this. Also, we are starting to deploy the mid-density genotyping service in pre-breeding. It's a project funded by the Crops to Entanga initiative. And what we do here is my last slide. So we are trying to, in a more recurrent genomic selection setup, integrate QTL, especially for two diseases, spot blotch and septoria twittici blotch, that are two diseases that are very polygenic. So we do not know many large effect QTL, and we are trying to, in a specific crossing scheme, so crossing these with elite lines, applying a recurrent genomic, rapid cycle recurrent genomic selection on top of it, crossing again with elite, so cycling these a few times to constantly integrate this polygenic QTL into our elite pools. With this, I want to stretch again. This is the work I showed. This is an absolute team effort. So we work a lot with the trade specialists, certainly, and the breeders. And nothing would be happening without the support of our national staff. And I also want to thank certainly our main funders. And with this, I'm happy to have questions. Thank you. I think I stopped sharing.
Thank you, Susan, for the excellent presentation. Thank you, Kelly, for the presentation of Susan. So the floor is open for questions. And meantime, if nobody asks anything, I have one question for Kelly and one question for Susan. So Kelly, my question to you is, because I'm in Africa, it's related to Africa, this continent. So what are the opportunities for collaboration with Africa?
Yeah, I think that's a really good question, and thank you for asking that. So one of the things that we try to do is to partner with scientists from all over the world. So if there is an interest in a particular variety that would be specific to Africa, then we would, certainly try to incorporate that in our work. And the other aspect that is, one of the ways that people get involved with the International Wheat Genome Sequencing Consortium is that they decide they want to try to get funding for a particular project. And then we will endorse that and bring them into the IWGSC as part of our coordinating committee. One of the things, just to point out, we are focused on genomic tools. So if it is direct breeding, then it's probably more of a wheat initiative effort that you might seek support for, or even with CIMMYT, because they're the ones who are actually translating the work. But if it's to develop genomic tools or to get training on how to do genomic tools and resources, then yes, come, talk to us. We're happy to try to develop programs, and, certainly want people to understand how to use these tools. One component of the National Science Foundation grant is what's called a STEM, science, technology, education, or no, I don't remember what's right now what it stands for. That's the problem of getting older. But in any case, it's for educational opportunities. And so one of the things that we're doing is trying to design things that could be used by young people or, and our team is working on developing something, maybe even a book about the wheat genome, which could be used by people all around the world. And we certainly want to get more people involved in wheat genomics. So anything that we can do to help in an educational standpoint, we're very much for it. And Joshua Stein, who is one of our team members, is really the lead on this project, and is very involved in annotation and has extensive experience in annotation. And if there is an interest, we could set something up to where he would actually show people how to do annotation. And we've talked about some of those things. So those kinds of activities we're always open to try to do. And you don't have to just reach out to me. You can reach out to Suzanne as well and come up with, what do you want? What would you like for us to try to do? The other thing that we do, as I mentioned, we try to work with tool developers like Arbor Biosciences. We're trying to establish a new relationship with Thermo Fisher and the tools and resources that they have. And so we're very interested in what would be useful, and then try to get tool developers to actually prepare that and make that available.
Thank you, Kelly. So Rajneesh, after I can address Susan, Rajneesh, please just unmute yourself.
Okay, Susan, thank you very much. Really very deep presentation on this wheat crop, because I'm from the IITA, but I started my PhD on the wheat. So really this crop is in my heart, So for my question is this, now I look the work in the Asia and Africa, also in the East Africa. Do you think you want to move in the West Africa, something like IIT is there after this Ukraine and this? I think wheat can grow here well also in the West Africa part of this.
Yeah.
So do you think in the future, is there any plan for this year to move the CIMMYT Wheat Program in this region?
Yeah, definitely. I think probably Kevin on the call can explain that much better than I. But yes, the reason that why we moved one of our breeding pipelines to Kenya is really to see the potential of wheat in Africa, Ethiopia, especially Kenya, also a little bit West Africa, Zambia, etc. So this is currently heavily explored, and we're just putting together really a strategy. What would be the potential to make Africa more self-sufficient in terms of wheat production. On the molecular side, certainly we start working with Sridhar's group and Calvo. So we have done already some initial genotyping. So this has to settle, I guess, in the next few years once the breeding pipeline is fully established. But yes, definitely. Mm-hmm.
Can I ask one more question, if allowed?
Yeah.
So my next question is, so whatever we are seeing this, you are using the genomic assisted breeding and you are doing well. So what do you think for the future gene editing, to including the gene editing in this your research, to meeting the food demand for the future? Do you think is there any scope, or do you think the genomic is sufficient to do the future demand for the future? What is your view on that?
Yes, definitely. Certainly a little bit a complex question on the legal side still, but we have a project. So we have a full protocol established by our genetic resource unit at CIMMYT, so to gene edit wheat. But we've currently, I'm not aware that there is, we have a funded project, but we have a project where we align with the John Innes Center in the UK, where we will test gene-edited products in Kenya, Pakistan, and I think in Egypt. So these are rust-susceptible genes that were knocked out. So in terms of rust resistance, so nil lines are already developed, and I think this is going to be grown in Kenya, Pakistan, and Egypt the next 1 or 2 years. So WASP susceptibility gene is one component, and the second component is a gene related to nutritional, so to zinc and iron content, also in collaboration with the John Innes Center. So definitely there is, I think, as the legal situation gets better, certainly there's also patents involved in this, so this has to be sorted out. But as I know, I think there's also a CG initiative on gene editing that will hopefully support the center to make that happen. Mm-hmm.
Thank you. So, and I have one more, but I think you can go. I will ask later.
Mm-hmm.
Okay, thank you. So Susan, my question partially was already replied by you to Rajneesh, but what are the main challenges that your group face in the current research area, and how can we support more and better?
You mean in the aspect of traits or in the aspect of deployment?
Both. You can address both. Thank you.
Yeah. I mean, one of our biggest challenge on the trait side is certainly, I mean, the quantitative traits similar to each crop, right? Heat and drought is, climate change is hitting hard. I think we have to find ways to have faster progress in that direction. I think that is one of the biggest challenges. In wheat, certainly, wheat blast is one of these upcoming diseases, and we have only one resistant genes, functional resistant gene. So there's an urgent need of discovery in that area, but it has been seen not to be easy. So there has been work on discovery, but so far there are only additional minor genes discovered, what we try to apply. WASP certainly, so all the diseases is an ongoing challenge. In terms of deployment, I think we have been slow in the terms of the so we need, I think the bioinformatics needs to be improved a little bit in, at least at CIMMYT. So we struggle at the moment to make full use of the whole genome sequences. So this is one challenge I have been raising to you for some while. Definitely, and certainly, yeah, I think that's all coming to my mind, really.
Well, it's already a lot. Thank you.
Yeah, we have still a lot to work on. Definitely.
So, Rajneesh, I don't know if you want to still ask something or if somebody else want to
Yes,
ask.
I can ask my last question. My last question on this, I hope you are using this drone or UAV on the phenotyping on this side. I have not seen that one in your piece, so I don't know how are you using. And one program I was looking online somewhere in the middle from the Google, and they say this with CIMMYT and CIMMYT, and when the CIMMYT is coming, I think they're working on the wheat. So can you put more insight on that one? What are you doing and what the
we're exploring the UAVs for sure. The problem we have here in Obregon is that we have a small airport very close. So we are not currently allowed to fly the drones on our most important plots. So our physiology has been working quite hard to make satellite data available, and I think these we are trying to use these now in combination with pedigree data to even at an earlier stage, at a F4 stage, so it's around 100,000 plots, very small plots, to provide the breeders information at that stage already. So that is kind of something we push quite a bit. Mm-hmm. Yeah.
Oh, satellite data.
But also there has been, sorry, just to add, there has been in this project we have in our South Asian network, we do also work on adding additionally UAV data across the different our different target population of environments. So BISA sites, for example, we have BISA in India. So there we are additionally working to bringing this data, UAV data together from Mexico and different sites in South Asia at the moment, Africa not yet. Mm-hmm.
So my question on the satellite data is enough resolution is there to look the disease and something, how there is the plant health, something like that, and something how the ear spike is coming, they are healthy or not, something is enough satellite data resolution is good, I don't know.
Yeah, I don't think we are there for the diseases yet, but more for the physiology traits like canopy temperature or NDVI. So this is, I think, optimized, not for all traits yet, because the resolution is certainly much lower. But this is currently what we have to work with, so that we have no choice.
And what the height you are using to fly the drone for this wheat crop? Height of the
Oh,
drone?
I'm not sure about that. Sorry, I'm not an expert in flying drones. But right now, here in our main yield testing sites, we cannot fly drones. So this is the difficulty we face, and it's just
thank you very much.
It's just a legal
Thank you.
difficulty. Yeah, sorry.
Thank you very much for the deep.
You're welcome.
Anybody else want to ask any other question to Susan or to Kelly? Please just raise your hand. And if not, I think we came to a conclusion. No, Rajnish, you want to ask? Please unmute.
Hand is lower. I'm doing the lower. So just click it. Sorry for that.
Go ahead. Okay, you are lowering the hand.
Okay, so thank you. I think we came to an end of one more seminar of this series. Thank you all for your time, for being with us. Thank you very much, Susan, for the great presentation. Thank you, Kelly, for your time, and very grateful for this opportunity. So hope to see you in the next seminar focused on maize. And the presentation will be presented by Georgina Eyemba from ISRA, that is the Senegalese Institute of Agricultural Research from Senegal. Thank you very much, and join us next time. Thank you.
Thank you.
Thank you, and a nice day, everybody.